Hello. Let's put these on. This is going to be a brief video that should help you put some things together about the heart and cardiovascular system in a way that is not organized just like your textbook is. You might remember from, Bio 101 or something like that, how embryonic development starts with a fertilized egg, which quickly divides into many, many little cells. Those are all omnipotent cells. Each one of those cells can become any other cell in the body. At some point, that quick burst of cell divisions uh, gets away from just being a sphere, starts spreading around, and makes another bigger hollow sphere, which is called gastrula. At that point, the cells that make the wall of gastrula are in three layers. The outside is ectoderm, the inside is endoderm, and in between those two layers is the medium layer, which is called mesoderm. At that point, the cells of the endoderm, ectoderm, and mesoderm are uh, pluripotent. They can become a whole bunch of different types of cells, but not all. So the ectoderm cells can become any type of skin cell or nerve cell or sensory cell. The cells of the endoderm can become any type of cell in the gastrointestinal system, respiratory system, urinary system, and the cells of the mesoderm can become any type of muscle cell or cardiovascular cell, which brings us to cardiovascular. Cardiovascular system is one of the very first to start developing in the developing embryo. Uh, the heart, what is going to become aorta and what is going to become uh, vena cava, develop very, very early in the embryo. At that point, at that early point, you might have a beating heart and an ovary or a testes, and nothing else. Everything else in the embryo is still completely undifferentiated. Maybe a little streak of protonerve cells that are going to become a nerve spinal cord one day. That's about it. There's no kidneys or livers or anything at that point. It's very, very early, and the heart already starts to beat because, as you know, those types of cells have autorhythmicity, ability to beat on their own. They don't need any kind of external stimulus. At that point, the rest of the cardiovascular system begins to develop. How does it do that? It follows various growth factors. Various cells in the periphery of the embryo are going to be secreting molecules that are going to diffuse through the embryo. And developing arteries and veins are going to follow the concentration gradients of those growth factors and grow into that direction. The same thing happens with nerves. They follow their own growth factors in order to build nerves. So at this early stage, uh, you have a very kind of diffuse embryo, and these, growth factors are being secreted, and some arteries and veins grow in that direction. And there's quite a lot of room for, I wouldn't call it error, but it's a room for variation. So let's say you have aorta developing and starting to arch, and you have the first big arteries branching off of aorta following those concentration gradients of those growth factors. And in most people, most mammals really, the first three arteries branching off of aortic arch are going to be the brachiocephalic trunk, the left common carotid, and the left subclavian. And but it doesn't have to happen. There are actually a lot of variations. Anywhere between 1 and 7 major arteries can branch off of aortic arch. And those are not pathologies. Most of those are not pathologies. Uh, you wouldn't even know. You can be an athlete, you can run marathons and never know unless somebody takes a chest x-ray or you undergo heart surgery or something like that, and then they see, oh, you only have brachiocephalic trunk and everything else branches off of it later. Or you have, six arteries going, left and right subclavian, left and right common carotid, and left and right brachiocephalic. You have all six independently, individually branching off of aortic arch, and all of those are variants, and those variations can be found in other places as well, and it's not uncommon. So when you look at your textbooks and lab manuals and atlases of anatomy, what you're going to see it everywhere in every organ, every picture there is going to be the most common situation. That doesn't mean it's the only one, and it doesn't mean it's the only one that's normal. There's always going to be some other variation. And if you look at the additional readings, I have posted several links to oddities, including nerves, which show variation exactly where they branch off, and arteries and veins, exactly where they branch off. And then um, and there's a whole bunch of variation, which is why I'm missing having the lab in person so we could open up a few cats, because every time we open up a few cats we see a lot of that variation. Opening up a cat, or if you're in medical school, the human cadaver is important. Yes, you get a smell of it, and different things feel different under your fingers than you expected them to be. Some things are harder or softer or crumblier than you expected. It doesn't look like on the pictures. But you also see that every organ is different color in different individual, different shades. Color of internal organs is invisible to selection. There's nothing to select for. There's absolutely no evolutionary reason for a liver to be that dark or light or whatnot. So it's going to be accidental. There's going to be random variation in color and shape and size and position of a lot of internal organs. And you miss that if you learn only from your atlases and textbooks, because they're going to give you the norm and they're not going to show you the variation. So every time we open up the cats in the lab, we find something that is not the norm. Usually it's one of those aortic arch situations where it's not three arteries, but let's say two, and then the students figure out which two are those. And where are the others? Where are the missing ones? Where are they branching off later on? Uh, about a year ago, we had a cat which had two renal veins and two renal arteries going into each kidney. So instead of a total of four vessels, there were a total of eight vessels. And we Googled it and we figured out that it happens in about 3% of the cats. Now, there are billions of cats on the planet, so 3% is a large number of cats that are pretty efficient hunters going around there, although they don't have the usual number of renal arteries and veins. So all that variation is not pathology. It's not sick. It is norm. It's just that that norm is never captured in your textbooks and lab manuals and atlases unless you dig into the literature of variation, which surgeons need to know. So when you open up a patient and it's something different from the textbook, you're not surprised and you can work around it. Okay, so those arteries and veins are going to follow those growth factors, and they're going to grow into the periphery. And they're all coming out of the mesoderm, right? And the arteries and veins are going to follow the same uh, growth factors and are going to grow in parallel. You're going to have an artery and a vein following those factors together. They're going to grow together, and you're going to all often, very, very often, mostly, especially those arteries and veins that go into the limbs, the arms and legs, the toes and fingers, they're going to be paired like that. They're not going to separate during development, and you're going to always have an artery and a vein snugly together. Sometimes it's one vein surrounded by seven arteries. Sometimes, there's other situations where at least one artery and one vein are going to go next to each other. So, renal artery is going to be right next to the renal vein, femoral artery right next to the femoral vein, brachial artery right next to the brachial vein. You have a lot of those things. Now, there are exceptions because veins are slower and less efficient, and they take up a lot more blood. About two-thirds of the blood is always in the veins. There are some extra veins in some places where you need to have more veins to pick up the blood from larger regions. One artery can shoot the blood into it, but you need more veins to evacuate all the blood away. So let's say great saphenous vein. There's no such thing as a great saphenous artery. It branches off uh, and picks up the blood from the you know medial superior part of the leg, of the thigh. So uh, circle of Willis. It's just a bunch of arteries. There are no veins right next to them. So there are exceptions to this. One of the things that is very, very good about having arteries and veins snugly next to each other is in warm-blooded animals, any kind of animals, as a mechanism for conserving energy. So you have very, very hot blood in your heart, and the heart beats, and that hot blood goes into the aorta, and from there it branches into a bunch of arteries, and it's a very, very nice, hot, wonderfully warm blood. It took some energy, right? You had to eat, get calories in, and transform those calories into food, into ATP, in order to warm up all those cells that are going to warm up the blood so it's nice and warm. Uh, and now that blood is going into the periphery and is on the surface of your arm. And your arm is out in cold weather, or you're swimming, so you're in cold water, which really quickly takes heat away from you. You're losing energy that way. And so by the time that blood gets all the way to the periphery, it's going to be cooled off. It's going to dissipate some of that heat energy into the environment. It's lost for you. You would have to make up for it by eating more. Which right now that means buying more hair steer, but with our ancestors or other animals that means more hunting, and that means that in some years you're not going to be able to hunt enough, and you're going to die of starvation. You're not going to have any progeny, but your cousin who has a better cardiovascular system is going to survive and have progeny. So there is strong selection for arteries and veins to hug each other. Because why? Because if your nicely warm hot blood is going out of the arteries into periphery, and on the way there it meets the vein, and that vein is bringing blood back from the periphery. That's the cold blood bringing back from the periphery. If those arteries and veins are snug next to each other, the heat is going to transfer from the hot artery into the cold vein, which means as it keeps going and releasing more and more heat from the artery to the vein, at the end, tip of the finger or something like that, that arterial blood is going to be pretty cold. And it's not going to lose that heat into the environment. Instead, it gave that heat to the cold blood of the vein. So by the time that the vein brings the blood all the way to the heart, it's already warm. So the body doesn't have to generate more heat, doesn't need more energy from food in order to rewarm that cold blood that came from the periphery. This mechanism, and uh, I gave you some links, and I gave you a little handout, probably in a couple of places in Sakai, about countercurrent exchanges. This exchange of heat, where a little bit, the difference between the artery and vein at any given point is going to be small, but enough for a little heat to move from the artery to the vein. And as you go further, the actual values in Celsius are going to be different, but there's always going to be a small difference. So read that, think about it, watch the videos, because it's a very, very important process, because it's not happening just in temperature and just in us. It's very important in, let's say, birds that spend most of their lives standing in water, like flamingos, right? They would have to eat 10 times more shrimp in order to keep themselves warm, but their legs are very, very cold because of current transfer of heat energy from the arteries into the veins. Whales, those whales that little shrimps and stuff like that. They go through the water and open up those big mouths and go and grab a whole bunch of water. Now that's ocean, that's cold water. And so this countercurrent system is happening in their tongues. Their tongues are actually very cold, so they're not to radiate all that heat energy into the water, because then they would have to eat much more plankton than they normally do. And so this is a way to save energy. Understanding the logic of the countercurrent exchange is really, really important. And the countercurrent exchange of heat between arteries and veins is the easiest to understand. It's the easiest example to understand. So please look at those schematics, watch the videos to figure out how that happens, how the artery loses heat as it goes past the vein, and the vein gains heat because one gives it to another. The same thing happens in the respiratory system of the birds. The lung birds don't work like ours. You have blood going in one way or air going in the other way, and it's going to be a countercurrent exchange of oxygen, which is why bird eggs and well, that also means dinosaur eggs, not eggs, the bird lungs and the dinosaur lungs are much more efficient. They can extract much more oxygen than the mammalian wings can, which is why birds can fly and dinosaurs could run around with those big bodies. Uh, and you need to know this because at the end of the semester, we are going to get to the urinary system. And kidney, the loop of Henle in the kidney operates as a countercurrent exchanger. But there it gets complicated. If you did not learn and understand really how countercurrent exchange works in the arteries and veins and in the bird lungs, it's going to be very, very difficult to understand how the kidney does it because it has a double countercurrent exchange. The descending and the ascending loops of Henle each has its own, and they work together. So it gets really, really complicated. Sometimes you have to take physiology three times from three different professors before you really understand it. But I hope you do understand finally in the end how countercurrent exchange in the kidney works. And that's why you need to start now with these simpler, easier examples: heat exchange in the arteries and veins, and gas exchange in the bird lungs, in order to understand the kidney later on. So I hope you found this little mini lecture interesting. It's outside of your textbook, although a lot of these elements are kind of mentioned briefly in your textbook, but they're not put together. And I hope this putting together is going to help you understand the cardiovascular system better. We will meet for a review session on Wednesday, and any other questions on any other topic are welcome. I might make a couple more videos on other aspects of the heart and vessels soon. Thank you.